Well, good evening, ladies and gentlemen. It is 7 o'clock, and it is my delight to welcome you to the fourth of our 5 Spring 2022 Science for Communities lecture series. My name is Tom Miller, and I have the honor and privilege of being the director of the Chesapeake Biological Lab. And we are really delighted to continue bringing you stories about our science and why it is important, as we have done now for over a decade in these lecture series. For this spring semester, we've decided to focus on stories about the urban ocean. For those of you who've attended previous lectures in this series, you'll know that I justified this theme because where human society affects the ocean most is in those coastal areas around the world's big cities and ports. And it is in those regions where we see the biggest impact, but also more optimistically, it is in those regions where we will likely see the biggest responses to any change in behavior that society adopts in the coming years. In this series to date, we've looked at an international program called the World Harbour Project, which sought to try and understand tertiary treatment of waste and of planting of oysters, particularly in New York Harbour. We've looked at the ways in which the shipping industry is responding to the threat of invasive species distributed into these urban oceans as a result, unintended result of animals from ballast water or animals that colonize the sides of ships, and some of the technologies that we're exploring to see how we can control that risk. Last week, we had a wonderful story about metals in the urban oceans, and our colleague Andrew Hayes wove a story of his own childhood on the Great Lakes in Hamilton, Ontario, and the problem that its harbour faced, to the challenges of Baltimore Harbour with legacy contaminants of metals from their rich history of industrialized metal fabrication over many centuries. Next week, we will change direction quite markedly for this series, and we are looking forward to introducing Dr. Samir Kirshner, who is an urban planner and architect from Morgan State University, who has spent her career looking at how society should plan cities of the future and renovate existing cities to bring a closer connection between the people who live in those cities and the harbors that serve them. And I encourage you to join us again next week for that lecture. But tonight, we're going to turn to one of the biggest challenges that face the urban ocean, and that is the lack of oxygen in its waters. That lack of oxygen is caused by numerous different processes, and that will be the focus of tonight's presentation. Tonight's speaker is my colleague, Dr. Jeremy Testa, who is an associate professor here at the Chesapeake Biological Lab. Jeremy got his undergraduate degree from the State University of New York's College of Environmental Science and Forestry, where I am told mighty oaks from little acorns grow. And from there, he moved and became a mighty oak, taking graduate work in our graduate program at our sister lab, Horn Point, where he received a master's degree and a PhD. Following his PhD, we were lucky enough to recruit him to CBL. He joined our faculty in 2013 as an assistant professor and was promoted in 2019 to an associate professor. Jeremy and his students and colleagues focused their work on understanding interactions between the biology, chemistry, and physics that affect the coastal ocean. And in many ways, his work is a throwback to the interests of our founding director, Dr. Reginald Truitt, who understood that to understand any part of the Chesapeake Bay ecosystem. It is very much an interdisciplinary challenge. So I'm going to invite Dr. Testa to give his presentation. I'm going to remind everyone in the audience that because this is a Zoom conference rather than a Zoom meeting, it is not possible for us to unmute you to ask any questions you have about this evening's presentation. But we do encourage you to type any questions in the chat box. And then at the end of Dr. Testa's presentation, I will mediate questions by combining and sort of drawing themes together to try and explore some of the points that he raises in his seminar. And so with that, it is my utmost pleasure to introduce my colleague, Dr. Jeremy Testa, who's going to talk about oxygen, the keystone element. Jeremy, over to you.
Thank you, Tom, and welcome everyone. Thank you for coming and joining us this evening. As Tom indicated, and as my title suggests, I'm going to be talking about oxygen tonight and kind of focusing on this idea of thinking about oxygen as a keystone molecule. And it's always kind of an interesting challenge to talk about oxygen in estuarine waters because it's something that we can't smell, we can't see, we can't touch, but we all have an intuitive understanding of its importance, and it's really just an essential component of ecosystems. And I'm going to try to spend my time with you tonight kind of explaining why that is and giving some examples from the urban world in particular, and how oxygen's change in those environments and what it can teach us about our potential to continue to be on a restoration path in the coastal zone. So I use this concept of the keystone molecule, and for those of you who have had some ecology, I'm making the comparison to a keystone species, right? Like a species of plant or animal that has a major impact on its ecosystem and is really considered a essential part of that system that really all of the other parts of the ecosystem really kind of rely on. And perhaps one that can have really dramatic impacts on the ecosystem by its behavior and may not be replaceable by another species. So I have a picture of a beaver here, and thinking about a beaver that eats plants and builds these dams, changes the entire hydrology of watersheds. And I bring this up because I think we can think about oxygen in a similar way when we think about how coastal ecosystems work. So why do I make this point? What's the meaning? So oxygen is a byproduct of photosynthesis, right? This fundamental process that generates organic carbon that feeds all of the food webs. And then oxygen is also consumed by all the metabolic activity of all the organisms in the ecosystem as well. And as we'll talk about, when that metabolic activity is perhaps too extensive, the rate of oxygen consumption is so rapid that it depletes oxygen faster than it can be replenished, and you get oxygen depleted zones. And that's a problem because all metazoan or sort of higher life requires oxygen for their physiology. So it's one of the reasons we're quite interested in it. Oxygen also controls, or at least highly influences, a ton of chemical reactions in the environment, including how organic matter is broken down, how the nutrients that we typically think as fertilizer in ecosystems get cycled and recycled, and a whole host of other important dissolved materials. So it's really a key element there. It's impacted by biology, it impacts biology, but it's also really highly influenced by the physical movement of water. So changes in climate, changes in weather really can have a big influence on oxygen. And oxygen is also really sensitive to nutrient pollution. So it's one of the major targets for the type of restoration that we do in coastal ecosystems. Now, I wanted to take a moment to do a little drawing just to kind of give a perspective on just how extensive the role of oxygen is in the coastal zone. So if you'll bear with me, I'm not the greatest artist, but let's imagine we have an estuarine water here and we have an underlying sediment in that water. And we have dissolved oxygen, right? So, oh, I didn't mean to do that, but now I can do that, okay. So first we can think of all the microscopic algae that are in the water, and their photosynthesis produces oxygen. Their respiration also consumes oxygen, and so they're really kind of a key element of the ecosystem. Now, the organisms that live in the water, like fish, like zooplankton, microscopic or not microscopic, but small organisms, they also consume oxygen. Now, this organic material, like we'll talk about, produced by algae, sinks to the bottom eventually, and some of it comes in from outside, and that material is broken down by bacteria to consume oxygen as well. So oxygen is consumed as it sinks through the water, and as it's broken down in the sediments, it's also consumed by oxygen. Now, the physical movement of water also is relevant here. So if you have big inputs of water coming in or out with the tide, for example, or with other sources, it can bring oxygen in and out of the water. One of the major sources of oxygen for ecosystems is just exchanging it with that which is in the atmosphere. And if you get really windy conditions, that really can really enhance that exchange of oxygen with the atmosphere. And then oxygen can also be produced by other organisms. So it's not just the algae. You know, if you have grasses growing in the water or microscopic algae, these are also producing oxygen. And although I've alluded to it a couple of times here, perhaps the last relevant part of this story is that when you have nutrients coming into the system, that stimulates the growth of algae, as you've probably heard about in my past lectures or others, making that algal pool bigger, shunting more and more of that algae to the bottom water. And then ultimately, when that material is broken down, depleting the oxygen that's in the bottom water. And that really becomes the time when we really start to be concerned about what's happening. So you may or may not have heard this term hypoxia, which we use to describe the condition of low oxygen water. If you Google this term, you'll hear a lot about the medical sort of definition of hypoxia and how it's applied to our own health. But you'll also notice that it peaks up here, you know, when we think about oxygen deficiency in the biotic environment. And so there's a lot of different terms we use for this. One of the terms is dead zone, although we do know that some organisms can happily live in very low oxygen environments. So dead zone is somewhat of a misnomer. Now, we care about this low oxygen, as I've said, because organisms need it. And this is just one example of how we might diagram that. So this bar here represents the amount of oxygen in the water on a scale that we commonly use, milligrams per liter. And different organisms have different sensitivities to low oxygen. So, you know, striped bass and many of the fish and shellfish that we often think about are somewhat resilient, or they're tolerant of oxygen conditions that, you know, are in the 6 to 5 range. But as you get into this much lower range, 2 to 1, fewer and fewer animals can inhabit those environments. But bacteria are sort of just flourishing there. So they're always there to break down organic material. And so that's why when we start getting these really low concentrations, that's when we start thinking about problems. And in a place like Chesapeake Bay, which most of us live around, we have this area of the bay in the bottom water that goes oxygen depleted in the summertime. And so this is just one type of hypoxia that we have in Chesapeake Bay, where oxygen is typically depleted in April or May, and it stays low in this central, deeper part of the bay for most of the summer before being replenished in the fall. But that's just one type of hypoxia that happens in a big estuary. And what I'm going to try to talk about today is some of the other sort of modes of low oxygen water that develop, especially in the urban environment. And I just want to point out that, although low oxygen conditions can be something that's organisms can deal with, they can run away from, they can tolerate for brief amounts of time, maybe to go get some food that's easy to get to in the low oxygen zone. When oxygen gets really, really low, you can have effects that are quite dramatic, whether it's just being, the mass die-off of organisms, or in this case, in this box to the right, a bunch of organisms that are fleeing those low oxygen conditions that are running into the shallows, in this case, the extreme shallows, piling up on one another. And in Mobile Bay, Alabama, they used to call these events Jubilees because people used to show up with their bushel baskets and collect all the crabs and flounder and whatever else was getting pushed up to the surface and have a big party as a result because it was easy catching. So although it may have a short-term benefit, the long-term effects of this problem can be somewhat more severe. Now, low oxygen waters have been sort of increasingly well documented, I'll say, over time and over space. And this is a map that's a bit dated at this point, but makes a couple important points. First, what you're looking at, I should say, is a map of the world where the redder or pinker the color, the more intense the human imprint on the watershed or the land is. And all the white circles are instances where low oxygen water or hypoxia has been reported. And what you might notice is that in places like North America and Europe, and in sort of the coast of China and Japan, there's a lot of white circles and there's a lot of red land. And this sort of represents this idea that there's low oxygen water in the vicinity of places where humans are having a large impact. In particular, one of the ways to have those large impacts are by concentrating our populations, creating a lot of waste, and that waste being discharged into the coastal zone. I'll come back to this because there's some other relevant points here. One of the reasons this has become a topic of my interest and many, many others is that if you look at this plot, this gives an time series, starting sort of before 1910, of the number of reported hypoxic sites globally. And I think this impresses upon you that these number of sites have been growing exponentially, perhaps because oxygen's become more of a problem as human populations have increased and our generation of nutrients and discharge to the coastal zone has increased, but also because we're probably looking for it a little bit more as we've recognized the problem and as the types of programs that are put in place to address this problem measure oxygen more and more. Now, one of the interesting things about this whole topic of low oxygen water is that there's a handful of places around the world that are just very well studied. Chesapeake Bay is one of them. And these are the big, let's say, hypoxic zones that generate a lot of interest. So there's just a couple of examples here. The Baltic Sea, one of the bigger sort of inland sea hypoxic zones in the world. The coast of Louisiana here, where the Mississippi River, draining half the continental US, discharges its nutrients. The coast of the northwest coast of North America, on the coast of Oregon and Washington, where there's a dead zone, sort of in a place where this upwelling of really deep ocean water that tends to be low in oxygen, or at least carries a lot of nutrients that stimulate this algal growth occurs. And so there's a number of these around the world that we've studied very well, and that have very long-lived low oxygen zones. Baltic hypoxia can last for years. The oxygen on the low oxygen water on the Gulf can last for months at a time. But this sort of, these zones are somewhat different than the more urban estuarine zones, which have been fairly well studied, but have somewhat different of a profile of low oxygen water. So I want to turn and sort of focus some energy on these urban zones. Perhaps you've seen a map like this in some of the seminars this spring, but this is just a map of population centers in the world, and you can kind of see what you already know are, where the big cities are, the large cities or the mega cities in particular. And one of the interesting things about cities is that the bigger cities in most cases tend to be connected to water in some place, oftentimes coastal water, and maybe even more oftentimes water that's somewhat protected and perhaps not as big as these big estuarine zones I was talking about. And so there's a high potential that all this human impact gets kind of concentrated in a relatively small body of water. So I'm just giving a couple of examples here where you can consider sort of these canals and nearshore waters in Copenhagen or the inner harbor of Baltimore, or all of the water that's in and around the city of Boston. And these are just examples. There's dozens more of them around the world, but places where we know that low oxygen conditions have existed in the past, and that we've learned a little bit about how these type of systems respond. And so to build on this idea of how kind of these urban zones might contrast from the bigger hypoxic zones, I just want to sort of contrast them together. So if you focus your attention sort of on the right panel here, what I'm calling deep stratified estuaries. And stratified estuaries are places where freshwater and saltwater are both present, and the freshwater, because it's less dense, sits atop the saltier water, and you tend to get this, what we call a pyknocline, that kind of separates those two layers. And so when we discharge nutrients into these systems, this is a pretty common example of expressing what the main part of Chesapeake Bay looks like. Those nutrients stimulate algae that ends up in deeper waters, and the breakdown of that material consumes oxygen. Now, as we transition to thinking about urban estuaries that might be dominated by wastewater inputs coming from large treatment plants, the ways in which oxygen depleted are somewhat different. So for example, what we call labile organic material that breaks down readily and therefore consumes oxygen readily, is in abundance coming out of wastewater treatment plants. And that material consumes oxygen and lots of it. These wastewater treatment plants also discharge nitrogen, and the form of nitrogen that is released when organic matter is breaking down initially is something we call ammonia. That's this NH4 here. And when that molecule is in the presence of oxygen, let's say in a nearby water body, it reacts with oxygen and consumes it to produce the other primary dissolved form of nitrogen, which is called nitrate. And so this process, which we call nitrification in general, is another potential source of oxygen uptake. And so this is a way that we can sort of contrast these urban estuaries from other types of estuaries. Excuse me for 1 minute. Okay. Now, we've considered many ways in which we can think about the ways in which ecosystems degrade and how we can restore them as a consequence of the types of pollutants that we put in water bodies. And so I just want to give you an example of one of them. And so the idea behind this graph is that as we increase nutrient load, which is on this axis, and we consider the intensity of hypoxia, which is on the other one, as more nutrients come into the system, we get more hypoxia, this kind of degradation trajectory, if that's what you want to call it. And really, in all of our watershed management programs aimed to reduce nutrients, we kind of have this perspective that, well, if you can reduce those nutrient loads, we'll go back down the same path we came in this restoration trajectory. And we can kind of think of this as a linear recovery, where the system kind of can be brought back to what it was simply by reducing the pollutant that was previously put there. Now, this is probably an oversimplification of how the world works. And we certainly have plenty of examples where this isn't the case. And so here's just a number of different examples that people have hypothesized to sort of relate these things to one another. And so you can see here that there's a number of different relevant trajectories. So for example, I'll just show a couple. The nutrient load might happen for some time. I should get a color here that works. And initially, the system doesn't show any sign of degrading. but there's some sort of threshold that's crossed, and all of a sudden the system degrades rapidly without much more new nutrient input. And perhaps there's something fundamental about that threshold. Maybe it's just something about the depth of the estuary. And so when we start reducing nutrients, then maybe we'll just come right back down the way we came. But maybe that sort of threshold process exists, and maybe the system has just changed because of that pollution. And it's just not the same system that it was before we started polluting it. And now, even though you reduce pollution, the system doesn't really recover. It takes a little while to recover from that, ultimately coming back down in that system, but it takes a little bit of time. So this is kind of a framework in which we can kind of hypothesize what we expect might happen in estuaries in general as they respond to increases and decreases in nutrients, but also in particular can be applied to how we think things change in the urban zone. So I want to transition now and just give some examples, go on a little bit of a world tour and give you an impression of what we learned about low oxygen conditions in these type of places. So we're going to start, and I'm not doing this because Dr. Miller is English, but we'll start in the Thames. And this is a relatively old paper, but they were looking at oxygen conditions in the inner Thames in and around London. And they were trying to understand how improvements in even just some simple wastewater treatment plant changes change the oxygen condition in this part of the estuary. And so here's a graph that kind of represents what happened. And we're starting in 1955, and this is only going to go up to about 1980. Okay? And so what's being plotted here are two things: the pollutant and the relative oxygen concentration. So BOD is an acronym that'll come up a couple of times in these next slides, which is something we call sort of biochemical oxygen demand. And it's a measure of how much material is in that water that's going to consume oxygen. And so the load or the input to the estuary in the 1950s was really high and sort of gradually went down over time. Now the oxygen, the measure of oxygen we're using is just sort of a measure of the how much of what could be in the water is there. We call that percent saturation. And that sort of only went up a little bit and then hit some threshold point where it just went up really, really high. And I say really high, but it's only still at about 40 or 50% of what it could hold. But the pollutant went down and the oxygen came up. And if you put it on this kind of trajectory diagram that I just showed you, where now you're taking the two variables and relating them together. So this oxygen load, right, is what we had called BOD here. And then the oxygen deficit, which is what we're talking about with saturation. And so what you're going to do here is you're going to start in 1955 and you're going to move through time. And so at first there was this really big reduction in load, but the oxygen was really the same for the most part. And by the early 1970s, the oxygen started to come up somewhat. And then by the 1980s came up pretty quickly. And so this kind of looks like this threshold type of recovery that I alluded to in the slides previously. And I don't know that there was a clear reason why this particular path existed, but maybe the important thing is after some sustained reductions in the loading of material that would consume oxygen, those oxygen concentrations eventually came back up. So the next place we're going to stop on our tours, we're going to go across the North Sea, and we're going to go to the Skelter, the Skeld estuary, which is primarily in the Netherlands. This is another estuary that's really highly impacted by human populations. You'll notice that Antwerp's here, but there's many big cities in this watershed. And so this is another place that's really been a focal point of what we've learned about how urban estuaries have changed. So here's another plot, but we're going to have slightly different variables on it. And so this is another time series, and this time we're going from 1965 just up past 2000, so it's a little bit more modern. And the important variables here are, we're again plotting like an O2 deficit, right? So really high numbers are bad. That means there's a big deficit from what oxygen should be. And then we have a variable called DIN, which is dissolved inorganic nitrogen. So it's a measure of that nitrogen load. And then we have that BOD variable. And what sort of maybe is obvious here is that the oxygen deficit went down, meaning the oxygen concentrations got better, and the nitrogen concentrations went down together. And so again, this was a place where wastewater treatment upgrades led to changes in the system. And if we come back down to this kind of diagram again, where we're relating, again, the pollutant on the X versus the measure of oxygen deficit, and we start in the 60s, we'll see that that deficit went up as the nitrogen went up, and then we turned the corner, or they turned the corner, sometimes in the early 70s, and we saw the oxygen come back down. And so this looks a lot more linear, kind of like that idealized point of view where the pollutant drove oxygen down, and then as you remove that pollutant, it went back in the other direction. And in this case, you'll notice another thing is that the BOD, that sort of, which is kind of a measure of the organic material getting discharged, its decline was much weaker than the decline in the nitrogen. And so this was definitely a place where the ammonium was really, really high and was driving so much nitrification that it depleted oxygen in the system. And I only say that because very rarely in the ocean or in estuaries do we see nitrification, that process, be a big player in the oxygen sort of accounting for the system. And in these special circumstances, in the urban estuaries where wastewater treatment loads are so high, we start to see that kind of dynamic play out. I think the last urban estuary on this tour is we'll come back to North America into the Delaware estuary and really the Delaware River estuary in the vicinity of Philadelphia. That's the Philadelphia skyline you're looking at here. And so we're going to review some work that was done in and around the Philadelphia area and downstream of it, and how those oxygen concentrations changed. So in this case, we're just looking at the dissolved oxygen concentration, not a deficit, just the concentration. And this paper managed to go back to 1880 via some clever reconstruction and found its way all the way up to about 2005. And I think the change here is pretty obvious. Oxygen was relatively high before we entered the 1900s, depleted pretty rapidly, was extremely low, almost non-existent in the late 50s and the early 1960s, and then recovered rapidly to get back up more or less where it was at the beginning of this record that was basically 100 years prior to that. If we overlay the nitrogen changes in this estuary that resulted, or that are associated with that, there's sort of two important pieces of information here. The first is that these squares here are the ammonium concentrations, which were relatively stable and then shot up to really high levels and then declined dramatically, right? And around the time that ammonium went down is when the oxygen started to turn around and come back up. But you'll notice that the nitrate, which is the other nitrogen form here, was sort of increasing steadily and then really got high as the ammonium went low. And so what's happening here is that, what's possibly happening here is that all of this ammonium, either from the wastewater treatment plant or within the estuary, is being nitrified. So you're losing that ammonium, but you're gaining nitrate. Another possibility is they started aerating the wastewater in the system. And so that nitrification reaction was actually happening in the sewage treatment plant. So you start discharging lots of nitrate, but you're not discharging near as much ammonium. And you're also probably filtering out some of the organic material. And so you have two of those potential sources of oxygen lost that are taking place here. So that's basically what we're trying to articulate here. So I've talked about maybe some old stories, maybe some really success stories. As Dr. Miller alluded to, this is one of the places in the world where humans have really been able to sort of reverse oxygen depletion problems really effectively and really kind of rapidly. It's one of the nice things about having a pipe. If it's expensive, but if you can clean up that pipe, you can clean up the estuary. I'm coming back to this map because you'll notice that there's not that many circles in the coastal waters around India, around Southeast Asia, around China. And you know that these places have developed rapidly. These cities have grown rapidly. And so if we had a proper accounting of the low oxygen zones, we would see a lot more white circles in this place too. And we know that some of these areas are getting more and more hypoxic in the past 20 or 30 years. Just now are some of the studies on those systems coming out. And so this map here really reflects a lack of accounting more than it reflects a lack of the low oxygen conditions itself. So the last part of this presentation I want to leave you with is to talk about a little bit of a different type of low oxygen condition. In part how it's played out in one wastewater treatment dominated system around here, and then to think about that similar type of oxygen in some other systems locally around here. So what I'm plotting here is sort of the maximum depth of an estuary where there's hypoxia and the duration in which that low oxygen occurs. And I bring this up because thinking back to these big low oxygen zones I talked about, right, which, Chesapeake Bay is one of them that tend to be stratified, the Baltic Sea, these sort of permanent oxygen minimum zones that might occur in some of the upwelling areas. Those places are really deep. But as we start to move down to the really shallow environments, many of which are these waters in the vicinity of these large cities, we start thinking of the duration of hypoxia occurring on a cycle that's much more frequent. And so it's a different type of oxygen where you just have a steady stream of pollutants coming in, continually degrading or consuming oxygen and just keeping the oxygen low kind of permanently, versus places where the oxygen is still varying around a bit, but the pollution is causing it to reach lower and lower concentrations when it makes those dives down. And I'll sort of illustrate that here. So one of the cool things about oxygen is that it just varies over the course of the day, kind of following the sunlight, as you might imagine. So this is just like one 24-hour period, right, with a nighttime, and then we have the daytime when the sun's out, and then again, the nighttime when the sun goes down. And so the oxygen concentration at night is going down. It's going down because there's no photosynthesis to produce the oxygen to keep it high. And so it's just all the oxygen consuming processes are taking over, and they're just bringing that oxygen down. When the lights come on during the day, the plants turn on, they start producing oxygen, oxygen goes up. And then by the time the sun is sort of waning and eventually goes out, we go back down to the oxygen being consumed overnight. So we kind of have this diel cycle. Diel really refers to this 24-hour cycle associated in most cases with the sun. And so this can happen where you get these minimum, either in the middle of the night or right before sunrise, these minimum conditions with oxygen. And the important thing about pollution is that if there's more algae growing in the water because there's more nutrients, this cycle gets bigger and bigger and more amplified. We have an example of that. So the Back River, it's been in the news recently, you may have noticed, because the wastewater treatment plant there has been mismanaged. But this is a place where there's been really substantial efforts to clean up the pollution from that particular wastewater treatment plant. And if you've driven over the bridge nearby, you might have seen these digesters that are part of the plant. It's a really giant plant that services a significant part of Baltimore and its metropolis. And it discharges into the Back River right about here. So this is a little shallow estuary that's just received substantial wastewater loads for a long time until recently. And so what I'm showing you here is a plot of the inputs of nitrogen from this wastewater treatment plant in the Back River to the estuary itself. And this record just goes from 1985 to 2018. It's pretty recent. And maybe the most important thing to look at in this plot is the TN, which is the total nitrogen. It's both the organic kind of solid form and the dissolved inorganic form. And that nitrogen has more or less gone down over time, over this past sort of 35 years or so. And interestingly, we have some records of dissolved oxygen, which I'm going to idealize here in several of these periods. So if we start at a time when the loads were relatively high, just around the time what we call BNR, or biological nitrogen removal, was starting to happen in the plants, where they're discharging less nitrogen, you can see this relatively dramatic diel curve of oxygen that was observed in the system. And although it didn't always get hypoxic, it did get quite low. And so there's this potential for this nighttime oxygen to take place. As we move through time and that biological nitrogen removal process really started to take hold and was consistently implemented, we see that kind of a contraction in that daily cycle. And so the likelihood of low oxygen happening got much less. And then in around 2017, something called enhanced nitrogen removal, an even better way of cleaning up the wastewater, was put into place. And we had an even more contracted oxygen cycle in the system. So we can see how just even over the course of 10 to 20 years, by reducing nutrient loads to this urban estuary, the potential for these oxygen depletion events to happen really went down pretty dramatically. And so this happens in a lot of other places. So I just want to show you a couple cool examples. The Corsica River, which is near Centerville on the eastern shore of Maryland, and Pepper Creek, which is kind of a dead-end canal coming off some of the Delaware inland bays over here on the Delmarva Peninsula. And these are two places where this diel cycling happens. And it's not really that they're urban, but they are influenced, at least in Centerville's case, by sewage treatment plants. And they kind of give a good impression of how this works. So here's an example of just over 4 days in the Corsica River estuary. And the gray here is the nighttime periods, and the orange is the daylight periods. And you can really see the cycle where oxygen comes down until the sun comes out, and then it screams back up again. It goes down at night, screams back up again, goes down at night. And then actually what happened here is that it was a cloudy day and it just didn't come up as much. Next day, the oxygen goes down and starts to hit this point where we call hypoxic. And it actually stays hypoxic for most of this day where the clouds persisted. And so, when you're out there on a cloudy day and you're thinking how miserable it is outside and how you wish it were a nice, bright, sunny day, one of the nice things these bright, sunny days are is enough photosynthesis to happen to sort of keep the oxygen in these estuaries a little bit higher than it might otherwise be. And so if you just look at how this plays out over the whole course of a year, you can just see how extreme the oxygen varies. And once you get into the summer months, May to October, you can see that the oxygen is sort of routinely dipping below that 2 milligram per liter threshold we commonly call hypoxia. Okay, so the last example I give you is from Pepper Creek, and this is from some of my work by my friend Damian Brady. And this is an even cooler example of this diel cycling hypoxia, because he put enough sensors out all along this estuary here. You can see all these stations here. This is Pepper Creek, that you can really get an interesting picture of how this develops. I'm going to show you kind of a video to express that. And notice here that there's kind of a number of axes here, and I'll sort of walk you through it. And so there's time on one of these axes, and then there's distance from the head of tide, which is kind of upstream to downstream. So low distance from head of tide is upstream, and then you're sort of moving yourself downstream. And then the height of these is the dissolved oxygen. And I'm going to erase this so you can see the video okay. Right when the good stuff was going to happen. We'll get back to it. Okay. So I'm going to sort of walk you through this. So we're going to twist this figure around a little bit. We'll take a pause, And so now what you're looking at is that distance from head of tide, right? So upstream is on the left, downstream is on the right, and we're going up through time. So day 189 to 198, we're working with about 9 days here. And the black here is the really, really low oxygen water. So you can see that as time goes on, that the oxygen starts to get low overnight, about halfway through this time series. And in a couple of these days, you really start to see that oxygen spread downstream. We'll let this twist around a little bit. Kind of see it in another view. So now the oxygen concentration is on this top axis here. And now we're just looking at time. And so you can see again how there's those really big swings every day, but they're not getting hypoxic, but eventually they start to dip down a little bit more. And by day 194, you're seeing those low oxygen concentrations really kind of persist. And so the last view of this will be to twist this around a little bit more and take a look at this over space. Oh, I lost it. Well, what you were going to see is that the low oxygen waters were most severe upstream, where the water movement and the residence time is really high, allowing plenty of time for that low oxygen water to deplete. So I'll end here. I just want to summarize by kind of reemphasizing a few points. The first is that oxygen is just this absolutely central molecule in aquatic environments. And so the depletion of it, because of that, is a global problem. It's a persistent problem, but it's one that's been solved, especially in urban environments where the sources of the pollution are relatively easy to control. And yeah, these urban estuaries have kind of unique features of low oxygen water. And our best examples of recovery for low oxygen waters worldwide exist in this urban zone, even though there's many growing ones around the world. And I won't have time to talk about it tonight, but the future for these type of environments is relatively uncertain. There's a lot of different factors that control oxygen, as I've shown you. And so you can imagine as the climate changes, as ecosystems change, as species move around, the picture for dissolved oxygen as we look into the future is relatively complex. So thank you for your time and your attention tonight. And I'd be happy to entertain any questions you have. Thank you.
Well, Jeremy, thank you very much. I will add one little anecdote myself to the story of the Thames. I grew up on the very eastern end of that map, just where London sort of ends. You'll see a little town called Erith, right sort of just below the Thames, right there. That's where I grew up. And in 1970, 1972, when I first started getting interested in fishing, no one would fish in the River Thames because there were no fish at that point. And I go back there now, and the waterfront at Erith is full of people with their rods and reels catching cod and catching flounder and catching a wide diversity of fish, including salmon that are now running back up the Thames again. So it really is a remarkable story of recovery, and I think a lesson to us all that if we're willing to take some of the hard choices, which are often economic choices, we really can see rapid responses. So with that, let me turn to the audience. And Len Zuza asks the question, what role does salinity play in the oxygen cycle? Is there some areas where salinity makes it more challenging to understand or less challenging, or it sort of change the dynamics in the oxygen cycle?
Salinity is certainly an important part of this. And, you know, I think there's a lot of small effects salinity can have. Like, for example, salinity has a small impact on the solubility of oxygen, you know, how much oxygen the water can hold. The most important way that salinity kind of directly affects this problem is that where you have a relatively salty environment that gets fresh water discharged into it, the potential is there for that fresh water to sort of layer over that saltier water and create this vertical separation of kind of a fresh surface water and a salty bottom water. And if there's no mixing happening there, which often there is not, there's two places water can really get oxygen for the most part, from photosynthesis or from the atmosphere. And so these dark, deeper waters that are sort of cut off from the surface water from this layering just don't have any source of oxygen. And they also accumulate the material that leads to oxygen depletion. So that's, salinity kind of creating the environment that allows for oxygen to be depleted without having a potential to be replenished is one of those ways.
All right, thank you, Jeremy. I'll go on to the next question. Yishan Lee tried to get away with asking 3 questions at once, so I'm just going to ask one of them to give time to other people. What's the role of climate change looking forward in terms of oxygen dynamics? Are they going to make this issue of hypoxia more or less relevant, and will they make solutions to it more or less complicated?
I think that there's a lot to that answer, but I think in short, if you ask most people who study this problem, they would tell you that at least in the temperate world, climate change is probably going to make this hypoxia problem more of a challenge. It's because warming tends to reduce the solubility of oxygen in the water. It tends to speed up the reaction rates that consume oxygen. We're in a part of the world where precipitation is likely going to increase or at least become more flashy. And so that's more river flow to create this separation of the surface and the bottom water, and it's more nutrients potentially being carried off the land. And so, like the Chesapeake Bay management plan that we have, the TMDL, is being adjusted right now to account for those climate changes. If we've looked at Chesapeake Bay, multiple people have looked at what Chesapeake, using models that represent the last 30 or 40 years, what would Chesapeake Bay have done in terms of oxygen in response to the nutrient loads we have achieved if there was no warming? Because there has been a warming in Chesapeake Bay since the mid-'80s. And the answer there is that the warming has sort of offset what we would have expected to have improvements for because of nutrients. Not 100%, but it's been a factor. And we would have expected the conditions to be better had that warming not existed. And I'll sort of leave it at that. I could talk all night about that problem.
All right, well, perhaps we'll have you back to talk all night about it at some point in the future. Jack Templeton asks, what is the prevalence of underwater grass, or does the prevalence of underwater grasses dramatically impact hypoxia?
I would say in most cases it does not, simply because the place, and it may depend where you are, but if we consider our local sort of mid-Atlantic environment and in the Northeast as well, the plants tend to grow in places that have a lot of light. And those places that have a lot of light typically don't have a lot of material in the water that is going to stimulate oxygen to be consumed. So they just tend to be in healthier places. The plants themselves produce oxygen. And so they almost sort of create their own sort of habitat in that way that's oxygenated. But there are cases where people have documented that where you have these grasses, if they're really dense and they really dominate the water column, just like algae, they can produce a lot of oxygen during the day, but then consume a lot of it at night. And so you might get some of these low oxygen excursion in some beds overnight. I myself haven't actually seen that, and I've worked in a few SAV communities where the oxygen concentrations actually remain quite high, perhaps because of the reasons that I sort of first answered this question.
All right, we'll take it. three more and then we'll call it quits. So the first of those three is from one of the attendees, an S. Schwartz. I don't know whether it's a Mr. Schwartz or a Miss Schwartz, but they ask how much of the bay hypoxia is attributed to circulation and seasonal stratification, therefore in a sense uncontrollable, and how much is it responsive or responsible for by nutrient loading and therefore controllable?
That's a really fantastic question. It's a really relevant question. It's one that's not all that easy to discern, to answer in some ways. The way, and in part, it's difficult to answer because the circulation, right, and the way that changes dynamically, and the way, the ability to measure oxygen frequently enough and have good enough information about the circulation and the loads is almost impossible to do with observations. We will never have enough money. We may not even have the right number of instruments to actually characterize the Chesapeake Bay well enough to answer that question. So the only tool we really have are numerical models that quantify this dynamic. And I think what the community, when I say we, has been able to kind of ascertain is that, and I don't want to put numbers on it, but there is a measurable component of it that is due to, let's just say, circulation and weather. Some studies have looked at, basically assumed that the amount of oxygen that is consumed in the water doesn't change at all. It's just the same. And that's important because with more nutrient loading, we would expect that oxygen consumption to be higher, but they just assumed it was the same, ran a model for the last 30 years, and just were able to kind of reproduce the volume of low oxygen water. And what really, the conclusion there was that, well, if you can get the stratification and circulation right, and you can get the wind mixing right, it doesn't really matter how much oxygen is being consumed. You're going to be able to get the hypoxia right. And so now the problem with that is that, now this is getting into a long answer, but if you change that oxygen consumption rate just a little, your sort of prediction of the hypoxic zone just changes dramatically. And that's because I think how much oxygen gets consumed kind of sets up these patterns of oxygen, and those patterns of oxygen where it's high in one place and low in the other determine how much the physical movement of the water is going to push oxygen around and either replenish it or not. And so the two things are just kind of inseparable in some ways. But I will say that the simulations that have been run, number one, that sort of in the model take nutrients out of the system, that the low oxygen zones can come down substantially. I would say at least more than 30%. And also, there have been some reductions in some measures of the low oxygen water in Chesapeake Bay over the past 30 years, despite warming, because the nutrient loads have been reduced. So we've seen that happen. And finally, we know that the circulation of the bay has not changed dramatically over the past 50 or 60 years. And the low oxygen volumes have gone up 2- or 3-fold. And there's no sufficient physical explanation for that I would argue. So maybe that's enough, and I'll leave it at that. But if you'd like to hear some more details, I can put my email in the chat for any, if you're willing, or you can Google my name and you'll find me. Thank you.
We can also take that attendee's details and put two of you in touch. So as always, good questions tend to elucidate long answers. So two short questions to end with. The first one will stay in the Chesapeake Bay, and Ted Karski asks, are there recent data for the Back River to demonstrate that nitrogen loads are increasing and oxygen is decreasing?
I'm going to assume that question is related to maybe the issues they've had with a wastewater treatment plant.
I would assume so too.
And there are measurements being made right now, but I haven't seen them. And we've actually, we've been looking at some data in the Patapsco just next door, which also has a big sewage treatment plant that's been having the same types of problems. Perhaps it's not so dominant to the estuary as the Back River plant, but at least up until the spring of 2021, it wasn't clear that there was a massive impact yet to the nutrient concentrations and the algae there. That's not to say it won't be revealed once we have the next year's worth of data, but the short answer for the Back River is that the data that really would capture up until now are just not quite available yet, even though they're being measured.
All right, and we'll end with one last question from John Overstrom. And we focused a lot tonight on human-induced causes of changes in oxygen concentration. And John says, you did not discuss whether there are naturally occurring dead zones, but it appears that the Pacific Northwest and the coastal ocean off of Oregon and Washington is a naturally occurring low oxygen region. So sort of two-part questions. Are there naturally occurring dead zones? Did they occur in the Chesapeake perhaps? And how extensive are naturally occurring low oxygen zones?
Oh, that's another great question. And, I think as you alluded, when we think about the zone off, the Pacific Northwest, that's a place where you have what we call upwelling. And I sort of described it, but it's kind of like a natural analog for sort of watershed-based nutrient pollution. So you get low, deep ocean water gets pushed up to the surface. It stimulates a big growth of algae, and as they sink, they are broken down and they consume oxygen. And maybe some of the water that's coming up with that upwelling is also low oxygen itself. So if you look at the coast off of Peru, off of Namibia, for example, the western sides of the continents that have these upwelling zones, because they have relatively narrow continental shelves, you see low oxygen zones happen in pretty much all those places. So they are an example of naturally occurring zones. The Baltic Sea has probably always had low oxygen water in it. The Black Sea is a huge portion of that deep water is practically anoxic, and it's likely always been that way, just because its circulation is just so restricted, because the only real outflow is through the that sort of narrow section through Turkey. So and Chesapeake Bay, there's indices that suggest that oxygen's been low in Chesapeake Bay, even at the time that the Europeans have arrived. The circulation of the bay is such that there's a part of the bay kind of right where the Bay Bridge is in that deeper channel that just kind of wants to be hypoxic. It's just set up to be hypoxic, and it probably to some extent always has been. What we've observed, over the past 70 years is just a great expansion. So thanks for your question.
All right, well, we'll end the questions there. We have recorded this lecture. We will post it to the website for the Science for Communities series in the next day or two. It will include all of the questions and all of the answers to it. And we will reach out to those people who ask questions that we didn't have time for and pose them to Jeremy, and I'm sure he'll take the time to send a brief answer back. I want to thank all of you for attending tonight. I know I always learn something from these evening lectures, and that was certainly the case tonight. And I want to encourage all of you to return here next Tuesday for a different take on urban oceans. One, we've had the last 4 urban oceans very much from the water viewpoint. And we're going to turn the tables next week and look at urban oceans from the land viewpoint as a planning and architecture challenge, and I'm really looking forward to that presentation. Samir Kirshner has done some amazing work around the world, including in some of the areas Jeremy's talked about tonight. And so it should be a great lecture. So Jeremy, once again, on behalf of all of the attendees, thank you very much for your presentation tonight. Thank you very much for your thoughtful answers to all the questions. And for those who attended, thank you very much for your time. And we hope to see you next week. Thanks very much indeed. Good night, everybody.
Good night.